Okay, good morning, everybody. My name's Sami Alrashed. I'm going to be presenting my project today. I just got to minimize this tab so I can read my actual project title, but it's uncovering activation and plasticity of tumor microenvironment in search for novel therapy approaches in GBM. So that's a mouthful, but hopefully it makes more sense at the end of my project, what that's supposed to mean. The first thing that I want to do is just acknowledge the Brain Tumor Foundation of Canada and the donors of the organization for actually allowing me to have this opportunity to go through the studentship and to actually be able to continue conducting research over the summer. Obviously, it's something that I'm really passionate about. So to be able to have the opportunity to keep doing research, even when I'm out of that field now, it really means a lot to me. So yeah, just thank you to the donors and also to all the staff and everybody involved with the organization. So just to introduce myself a little bit, I'm originally from Windsor, Ontario. I went to the University of Windsor. I graduated with a degree in biomedical health sciences there. And I was in Dr. Lisa Porter's cancer research lab since 2018. Now I'm a MD 2025 student at the University of Ottawa. And obviously, as I've already said, I've been doing research in the past summer as part of the studentship, and I'll also be doing it next summer in 2023. So I guess to sum up what the goal of this project is, and again, it'll probably make more sense as we go through. I'm looking at a type of brain cancer called glioblastoma, and I'm looking at another cell type in its like immediate environment that influences it. And I want to figure out how this cell type called fibroblasts influences the actual progression of the tumor. So with that being said, I'm just going to jump right in and give a little bit of an introduction. So glioblastoma is abbreviated as GBM. It is the most aggressive type of brain tumor. It's actually a grade 4 astrocytoma. An astrocyte, it's just a type of cell in the central nervous system. And grade 4 is unfortunately the worst grade in the grading system of cancers. So it is very aggressive, obviously. It has a very poor prognosis. Even with treatment with the standard of care, it's only around 15 months for median survival, which is quite low. And there's less than a 5% 5-year survival rate. Even though it's super aggressive, it doesn't mean that it's like a rare tumor or anything. It actually accounts for 17% of all brain tumors, and it composes 52% of malignant brain tumors. And these were stats that I got last year from a different presentation as well. So this might have even increased since then, unfortunately. But another thing about GBM is that it's a very heterogeneous at different levels. So nothing about it is uniform. And that makes it almost impossible to treat, or at least very difficult to treat, because you can't use the same approach for every single patient. Pretty much everybody's always going to require something different. If there was more than one treatment, somebody would require something different because of how unique each one is, pretty much at the genetic level. So in terms of treatment, I talked about that standard of care. It's actually a chemotherapeutic agent called temozolomide or TMZ. And that's typically used in combination with radiation therapy. So surgical resection or like surgical removal of a tumor is usually what you would aim for in like any kind of curative aspect of a cancer. But for GBM, since it's in the brain, a lot of times it can be in kind of like a critical area. So like, for example, not too sure what's immediately surrounding this area, but let's say there was like something related to speech or like comprehension in that area, then it would be a little bit of a risk, or not even, it would be quite a high risk to try to surgically resect that without even knowing 100% that you've fully removed the tumor. So that's why they tend to go for chemotherapy with radiation. And even then, really difficult because of that aspect of heterogeneity that I mentioned, where everything is kind of unique at the genetic level inside of the tumor. And also because of the blood-brain barrier, which is kind of like a moat around the brain that really prevents entry of molecules. And that includes even like good molecules, so like chemotherapeutic agents. So I think I'll reiterate that in this slide. Yeah, so this is what I was going for with that blood-brain barrier. even if you have a good chemotherapeutic agent, it doesn't mean that it's going to be able to cross. So that really limits the number of drugs that we have to treat GBM, unfortunately. And even if it can cross, it doesn't always go to the tumor site. Another aspect, this is kind of just retouching on that heterogeneity aspect. So inside of a tumor, you'll have kind of primitive cells that we call cancer stem cells, which is like this little pink dude in the diagram here. And these can give rise to other cancer stem cells, so they can kind of clone themselves, or they can give rise to like a cell that's going to become an adult cell, so differentiated, like this green one. And these are the ones that compose the majority of the tumor. So like the bulk of the tumor would be made up by these differentiated ones. Typically, when we treat, we're getting rid of these, and it keeps these ones alive, and that pretty much keeps them with the power of being able to just constantly regenerating or recreating or recapitulating the tumor. So the goal in therapies is trying to get rid of these cancer stem cells. And it's important to be able to figure out what kind of niche signaling these cancer stem cells have. You know, what cells are they interacting with, what signaling pathways or what molecules are impacting them, and allowing them to be able to go through that like bidirectional pathway of cloning themselves and of regenerating the tumor. And that makes them like really valuable in terms of future treatments, and trying to figure out like how can we block this molecularly. So this is just a small reiteration of everything I've said, with the whole idea being that the tumor isn't just an independent tumor that's doing everything on its own. It's in a microenvironment that has tons of things around it, and those are all influencing it. And it's not just interacting one at a time with each of those. Everything's all going on at the same time, and that's what really makes things difficult to treat. So in terms of my work, I'm not going to be presenting everything that I've done to date. We have some other work that we've done, but we really want to actually redo that next summer to make sure that the results are accurate, because they're a little bit interesting. So I'm going to focus on some other pieces of data today. And these ones were my objectives. So I wanted to optimize conditions for human glioma and fibroblast coculture. Glioma is just like the brain cancer cells that you can purchase commercially and then culture. Fibroblasts are a type of cell in that tumor microenvironment. They're actually involved in like wound healing in the normal body. And co-culture just means that they're cultured or grown together in the same plastic dish. Second thing was I wanted to use this fancy machine called the fluorescent activated cell sorter so that I could separate these into independent populations. So this machine lets you take like a clump of cells, for example, maybe there's 10 different types of cells, and you can use these lasers that detect features on the cells to basically split them up into independent populations, and then you can work with those populations, which is great. And then I wanted to determine transcriptional or genetic expression changes and changes in the basic biology of those glioma cells that were exposed to fibroblasts in a co-culture. So that's the data that I'm gonna just glance over. Oh, I have one more thing I want to mention. So when I say optimize conditions, I pretty much mean that I'm starting in 2D cultures, and then I'm working up my way up to 3D cultures, and then I would be working my way up to actual patient samples. You don't want to jump right in off the bat, for example, to patient samples, because it could be a waste of resources if you don't know pretty much like the experimental parameters and the best way to culture these cells. So the best thing to do is always start 2D, like this diagram here, just like a flat layer of cells on a plastic dish. And then you can work your way up to 3D, which is more like now you're starting to see little spheres floating, or you can apply like a matrix that creates a tumor microenvironment to make this 3D. And then you could do the same thing, but use patient lines in the future. It's really important to be able to use 3D cell culture, because it's much more representative of how things work in our body. Obviously, we're not two-dimensional inside, so it's very limited in terms of the accuracy of how these cells behave when you work in two-dimensional. Just as an example, in this diagram here, the cells in the center are going to be different than the cells on the periphery, because of things like what cells they're interacting with, what kind of diffusion level of oxygen or nutrients, or environmental stress that they might have in that region. That can all change how they behave genetically. So this is just a diagram of everything that I'm going to go over. It just kind of demonstrates here. I have the pink cells are fibroblasts and the green cells are glioma cells. This is a 2D culture, this is a 3D culture, and these are kind of just the experiments that I'm going to talk about. I won't spend too much time on this picture here, but it is a nice layout I can go back to later if there's any questions afterwards, or I can send this to anybody. So the first thing in terms of basic biology that I wanted to look at was the cell cycle profile of glioma cells that were cultured with fibroblasts. So when I have co-culture, that's what I mean, the ones that were exposed. And when I have monoculture, these were just control glioma cells that were never exposed to the same environment as a fibroblast. You can see in these two spots here that when they were exposed to fibroblasts previously, these cancer cells actually started to undergo more DNA replication, which kind of indicates that they're more active, and they underwent more mitosis, which is the M here in the dark green color. So they were dividing more and they were more active, and that corresponds with this next diagram over here, where we co-cultured glioma cells and fibroblasts at different densities of fibroblasts to see if the fibroblasts could influence the rate of proliferation or division of those glioma cells. So yellow here is fibroblast. This is the monoculture, and then the dark green, sorry, this is the co-culture, and then the dark green is the monoculture. And you can see there's different ratios. So for example, 1 to 3 here is for every, this just means that I have 30% fibroblasts, and this is 50% fibroblasts, and this would be 10% fibroblasts. And what we saw here is that when you culture them with fibroblasts, the glioma cells actually significantly proliferated more relative to that monoculture. So pretty much putting them in the same environment as fibroblasts led to them having a higher proliferation rate, and led to them being more active and undergoing more mitosis in terms of the cell cycle behavior. Transcriptional analysis or genetic expression analysis. This was a little bit interesting. We want to be able to redo this next summer with different models. I believe we were only able to do this with 2D. So it wasn't the way that we wanted it to be yet. But what you might see that stands out here is these two, SOX2 and YKL40 in the red, which represents co-culture, are overexpressed relative to the green, which is the monoculture. SOX2 is a really important regulator in terms of like stem cell fate, and allowing them to continue behaving as stem cells. And YKL40 is a marker of poor prognosis. So if you have higher expression of YKL40, then that tumor would be indicative of a worse prognosis. The rest of the results were a little bit interesting. They were supposed to be markers of tumor aggression and behavior, but the results weren't what we were expecting. So I don't really want to comment on those too much without confirming this later on in my future experiments in summer 2023. But kind of to go hand in hand with the SOX2 aspect of stem cell regulation, we did a 3D culture where you take, the cells and you put them in a dish as single cells. So it's kind of like it's in its own little island. And you want to see if that single cell has the ability to create a tumorigenic foci. So for example, this is one foci. And it's basically talking about the idea of being able to generate a tumor from one cell. So this is the monoculture on day 8 and day 12, and then this is the corresponding co-culture. You can see just from the pictures alone, let alone the data, that there's a lot more clonal growth in the co-culture than there is in the monoculture. So that was a really interesting result to see. It just keeps pointing at the relationship between fibroblasts and the glioma cells, or at least in terms of glioma cells being impacted by their tumor microenvironment. Something similar that we did in a 3D experiment was we wanted to see the growth rate of those tumors. So we fluorescently labeled them so that they would glow and we could kind of track how dense they were. And we measured that over a span of 19 days. And it seemed like the co-culture ones grew at a faster rate, and overall they had higher growth relative to the monoculture ones. So they were bigger at the end of these 19 days. But this is something that we have to repeat still, so we can see if it's actually statistically significant or not. So that is also going to be happening next summer. And then I think this is the last thing that I'm going to touch on in terms of experiments, but we wanted to see if culturing the glioma cells would impact how they responded to drug treatment. And again, the drug treatment would be with the standard of care, temozolomide, and that's going to be relative to a drug vehicle control, which is just this chemical known as DMSO. So on day 1 versus day 2 in the co-culture TMZ, you can see that the cells underwent growth. So it jumped from this value all the way up to here, and so did the co-culture DMSO. So this is showing us that there might be a relationship between the fibroblasts kind of helping or allowing the glioma cells to survive better in the same environment as the drug, or like in the presence of the drug. Because normally what you'd expect is that you would think the cell size would drop once you put the standard of care. So like as an example, this is in the monoculture. So this is the control, and then this is in the presence of temozolomide. The size of the tumor decreased because you put that chemotherapeutic agent, which is going to start killing off some of those cancer cells. But it increased in size here. We had an interesting result here. So we actually wanted to just see, we wanted to follow up, sorry, to see if we could confirm this aspect. So we did something called a cell viability assay, and that just looks at the viability, or like how many cells are living in that actual tumor. It pretty much breaks it up and it analyzes the viability of each cell, and then it spits out a number for like an overall representation of it. So this is the monoculture, and this is the co-culture. The blue is this DMSO, and then the pink is the drug, again, temozolomide. And you can see that for the co-culture, the cells that were exposed to fibroblasts previously, they actually have higher viability in the presence of temozolomide, or the standard of care, relative to the control, which you would normally expect to actually look like this, that they would have lower viability in the presence of the control, but it's the opposite. So that's something that's obviously going to instigate further investigation, because it is a valuable result to be seeing that one cell type can impact how another cell behaves to its standard of care. So that's something that is also going to be happening next summer. And yeah, I think that's everything. So just to summarize, I think it was a little bit sorry, kind of zoned out there. So to summarize the main findings, this was with the whole cell cycle thing: more division, more active, and they were dividing at a increased proliferation rate once they were in presence with fibroblasts, relative to cells that had never been cultured with fibroblasts before. That's both of these points. Exposing them to fibroblasts led to an increased expression of a stem cell regulator, as well as a marker for prognosis. Again, this is something that we want to look into more next year, because gene expression is really valuable in terms of studying the biology of a cancer, and it would be really important in terms of revealing what kind of interactions these cells might have with each other. When we did a little bit of 3D work, we saw that fibroblasts promoted the clonal growth, which was that ability to create those little like tumorigenic islands. And it also increased tumor formation rate, and the aspect of drug treatment that I just talked about, which is that the co-cultured cells were more viable in the presence of TMZ than the monoculture cells. And just to acknowledge again, thank you again to the Brain Tumor Foundation of Canada and to the donors. And also this is the Porter Lab. So thank you guys for letting me come back and to research over the summer, and to all of our labs, other donors and funders. We're obviously very grateful to have these resources and be able to conduct this research. If anybody has any questions regarding my presentation, feel free to contact anyone at the Brain Tumor Foundation of Canada, and they can try to answer your question. If not, I'd be glad to be put in contact with anybody and try to answer things on my own. So yeah, thanks for listening. That's my research, and hopefully I have an even more interesting update next summer.